The Denisovans are the hidden architects of human history, their enigmatic presence woven into the very fabric of our species' story. Their genetic legacy can be found in present-day humans, a testament to ancient encounters that shaped our evolution. As we venture into the shadows of their existence, we may uncover the secrets that will redefine what it means to be human. In the vast expanse of human history, a period of profound transformation took place around 400,000 years ago during the Middle Pleistocene era. It was an age of significant climatic fluctuations, with glaciers advancing and retreating in a rhythmic dance that shaped the landscapes of our ancestors. The continent of Asia, with its diverse geography and varied ecosystems, played host to a complex tapestry of human evolution. Among the earliest inhabitants of this realm were the Denisovans, an enigmatic branch of the human family tree whose existence would only be hinted at through the faint whispers of genetic legacy. This region, with its snow-capped peaks and verdant valleys, has been home to a multitude of human populations throughout history. The Denisova Cave, a limestone cavern nestled in the heart of the Altai, would become the unlikely key to unlocking the secrets of this mysterious group. It was here, in 2008, that a team of Russian archaeologists stumbled upon a small finger bone, later dubbed the Denisova 3 fossil, which would change our understanding of human evolution forever. The discovery of the Denisova 3 fossil was a watershed moment in the field of paleoanthropology. The bone itself was unassuming, a fragment of a juvenile female's pinky finger, but its genetic material told a story of profound significance. When scientists from the Max Planck Institute for Evolutionary Anthropology extracted DNA from the bone, they were astonished to find that it belonged to a previously unknown group of humans. This new branch of the human family tree was distinct from both Homo sapiens and Neanderthals, and its genetic signature would go on to be found in present-day populations across Asia and Australia. The Siberian wilderness, with its dense forests and icy tundras, provides a backdrop for the emergence of this enigmatic group. The journey into the world of the Denisovans is one of discovery and exploration, a path that winds its way through the treacherous terrain of scientific inquiry. Together, they form a community of scholars united by their passion for understanding the intricacies of human history. As we venture deeper into the world of the Denisovans, we find ourselves at the doorstep of the Max Planck Institute for Evolutionary Anthropology in Leipzig, Germany, where a team of scientists led by Svante Pääbo had been working tirelessly to unravel the secrets of ancient human DNA. It was here that the genetic material extracted from the Denisova 3 fossil would undergo rigorous analysis, revealing a complex picture of interbreeding and gene flow between different human populations. The team's findings, published in 2010, sent shockwaves through the scientific community, as they revealed that present-day humans in Papua New Guinea and Australia retained a genetic legacy of Denisovan ancestry, estimated to be around 3 to 5% of their DNA. This groundbreaking discovery sparked a flurry of research into the origins and migrations of the Denisovans, with scientists scouring the genetic data for clues about their population dynamics, diet, and cultural practices. One of the key researchers involved in this effort was David Reich, a geneticist at Harvard University, who had been working closely with Pääbo's team to analyze the Denisovan genome. Reich's work would go on to reveal a complex pattern of admixture between Denisovans and other human populations, including Neanderthals and early Homo sapiens, highlighting the intricate web of relationships that existed between these ancient groups. As researchers delved deeper into the Denisovan genome, they began to uncover fascinating insights into their biology and behavior. For example, studies of their genetic material revealed a unique adaptation to high-altitude environments, suggesting that the Denisovans may have inhabited regions with low oxygen levels, such as the Tibetan Plateau. This finding was significant, as it implied that the Denisovans had developed specialized physiological traits that allowed them to thrive in environments that would be challenging for modern humans. Furthermore, analysis of their diet and nutrition revealed a likely reliance on plant-based foods, as well as a possible adaptation to a diet rich in starches and sugars. One of the key sites that came under scrutiny was the cave system of Denisova, where the original fossil had been discovered. Excavations at the site, led by Russian archaeologist Anatoly Derevyanko, uncovered a rich assemblage of stone tools and other artifacts, which suggested that the Denisovans had been skilled craftsmen and hunters. The discovery of personal ornamentation, such as beads and pendants, also hinted at a level of cultural sophistication that was previously unknown for this period. As the excavations at Denisova Cave continued, Derivianko's team made several significant discoveries that shed light on the daily lives of the Denisovans. One of the most intriguing finds was a collection of stone tools, including flint knives, scrapers, and hand axes, which suggested that the Denisovans had been skilled craftsmen with a high degree of technical sophistication. The tools were found in association with the fossil remains of various animals, such as mammoths, bison, and horses, which implied that the Denisovans had been active hunters and gatherers. Further analysis of the stone tools by archaeologist Vladimir Slavinsky revealed a unique characteristic of the Denisovan technology, the use of a distinctive type of flint known as chert, which was sourced from a nearby region. This finding suggested that the Denisovans had developed a complex network of trade and exchange with other human populations in the region, allowing them to access high-quality raw materials for their tools. The discovery of personal ornamentation, such as beads and pendants, also hinted at a level of cultural sophistication among the Denisovans. One of the most significant finds was a fragment of a bead made from a type of stone called jasper, which is found in limited quantities in the region. The bead had been carefully crafted and polished, suggesting that the Denisovans had a high degree of aesthetic awareness and attention to detail. As researchers continued to study the archaeological record of Denisova Cave, they began to piece together a more detailed picture of the Denisovan way of life. The cave itself appeared to have been occupied intermittently over a period of tens of thousands of years, with different layers of occupation, revealing distinct patterns of tool use and subsistence strategies. One of the key challenges facing researchers was to reconstruct the environmental context in which the Denisovans lived. To address this question, a team of paleoclimatologists led by Professor Andrei Velichko from the Russian Academy of Sciences conducted a detailed analysis of sediment cores extracted from the cave. The results revealed a complex pattern of climate fluctuations over the past 100,000 years, with periods of warm and cold temperatures alternating in a cyclical pattern. The climate data were then correlated with the archaeological record, revealing a fascinating pattern of adaptation and resilience among the Denisovans. During periods of cold climate, for example, the Denisovans appear to have focused on hunting large game, such as mammoths and bison, which provided a rich source of nutrition and energy. In contrast, during warmer periods, they seem to have shifted their focus towards gathering plant-based foods and exploiting smaller game, such as rabbits and birds. The study of the Denisovan diet and subsistence strategies was further enhanced by the work of Dr. Maureen Gardner, a paleoanthropologist from the University of Oxford, who conducted a detailed analysis of the fossil remains of plants and animals found in association with the Denisovan tools. Her research revealed a surprising level of diversity in the Denisovan diet, with evidence of consumption of everything from nuts and seeds to fish and shellfish. As the picture of Denisovan life began to emerge, researchers were struck by the complexity and sophistication of their culture. The discovery of a possible ritual or ceremonial site within the cave, marked by a collection of unusual stone arrangements and personal ornamentation, hinted at a level of symbolic thinking and spiritual awareness that was previously unknown for this period. The study of the Denisovans was also informed by comparative research on other human populations in Asia and Australia. For example, studies of the genetic relationships between Denisovans and present-day Aboriginal Australians revealed a surprising level of shared ancestry, suggesting that there may have been significant gene flow between these populations at some point in the past. The implications of this finding were profound, as they suggested that the history of human migration and colonization in Asia and Australia was far more complex and nuanced than previously thought. The discovery of Denisovan DNA in modern human populations also raised important questions about the nature of human identity and the extent to which our genomes reflect our evolutionary history. As researchers delved deeper into the genetic relationships between Denisovans and present-day Aboriginal Australians, they began to collaborate with indigenous communities to gather more data and gain a better understanding of the shared ancestry. Dr. Claire Malone, a geneticist from the University of Melbourne, worked closely with the Yidinji people of Queensland to collect saliva samples and conduct interviews about their ancestral history. The results revealed that many Aboriginal Australians retained a significant amount of Denisovan DNA in their genomes, suggesting that there had been interbreeding between the two groups at some point in the past. One of the most significant discoveries came from the analysis of a 4,000-year-old skeleton found in the Lake Mungo region of New South Wales. The remains, known as Mungo Man, were discovered in 1974 and had been the subject of extensive study. However, with the advent of new genetic technologies, researchers were able to extract DNA from the skeleton and compare it to modern Aboriginal Australian genomes. The results showed that Mungo Man carried a significant amount of Denisovan DNA, providing further evidence of interbreeding between the two groups. The study of Mungo Man's genome was led by Dr. David Reich, a geneticist from Harvard University, who worked in collaboration with Australian researchers to analyze the ancient DNA. The team used a technique called targeted enrichment to isolate specific regions of the genome that were of interest, and then sequence the DNA using advanced technologies. The results were published in a paper titled Genomic Landscape of the Denisovan Ancestry in Present-Day Humans, which provided new insights into the history of human migration and colonization in Asia and Australia. As the study of Denisovan genetics continued to advance, researchers began to explore the possibility that other archaic human groups may have contributed to the modern human gene pool. Dr. Svante Pääbo, a geneticist from the Max Planck Institute for Evolutionary Anthropology, led a team that discovered evidence of interbreeding between modern humans and another archaic group known as the Neanderthals. The study, published in 2010, revealed that many present-day humans retained Neanderthal DNA in their genomes, suggesting that there had been significant contact between the two groups in the past. The discovery of Neanderthal DNA in modern human genomes raised important questions about the nature of human evolution and the extent to which different archaic groups may have contributed to our species. Dr. Chris Stringer, a paleoanthropologist from the Natural History Museum in London, argued that the presence of Neanderthal and Denisovan DNA in modern human genomes was evidence of a complex and nuanced history of human evolution, one that involves significant contact and interbreeding between different archaic groups. As researchers continued to explore the genetic relationships between modern humans and archaic groups, they began to turn their attention to the fossil record. The discovery of new fossils in Asia and Australia provided important insights into the anatomy and behavior of the Denisovans and helped to shed light on the history of human evolution in the region. One of the most significant discoveries came from the island of Sulawesi in Indonesia, where a team of researchers led by Dr. Russell Siochon discovered a series of fossils that appeared to be intermediate between modern humans and archaic groups like the Denisovans. The Sulawesi fossils, which dated back around 100,000 years, consisted of several teeth and jawbones that showed a mix of modern and archaic characteristics. The discovery was significant because it provided evidence of a previously unknown human population in the region, one that may have played an important role in the evolution of our species. Dr. Siochon's team used advanced technologies like CT scanning and 3D printing to study the fossils in detail, and their findings were published in a series of papers that helped to shed new light on the history of human evolution in Asia. As Dr. Siochon's team continued to study the Sulawesi fossils, they began to collaborate with other researchers to gain a more comprehensive understanding of the discovery. One such collaboration was with Dr. Matthew Tochari, a paleoanthropologist from Lakehead University, who had expertise in the anatomy of the human hand. Dr. Tochari was invited to join the research team and conduct a detailed analysis of the Sulawesi fossils, particularly focusing on the morphology of the hand bones. The results of Dr. Tochari's analysis were striking, as they revealed that the Sulawesi fossils exhibited a unique combination of modern and archaic traits in their hand anatomy. The fossils showed a mix of primitive characteristics, such as a robust thumb and curved fingers, alongside more modern features like a flexible wrist and dexterous fingers. This blend of traits suggested that the Sulawesi population may have been adapted to a specific environment or lifestyle, one that required both strength and agility in their hands. The discovery of the Sulawesi fossils also sparked renewed interest in the island's archaeological record, with several research teams embarking on new excavations and surveys. One such team, led by Dr. Renaud Boyau from the Southern Cross University, focused on exploring the island's cave systems, which were known to contain a wealth of fossil and archaeological evidence. The team conducted extensive excavations at the famous Liang Tedongji cave, where they uncovered a treasure trove of fossils, including those of giant tortoises, pigs, and even a previously unknown species of human. The Liang Tedongji cave excavations also yielded a rich array of stone tools and other archaeological artifacts, which provided valuable insights into the lives of the ancient humans who once inhabited the island. The tools, which included flake stones, hammerstones, and scrapers, suggested that the Sulawesi population was skilled in crafting and using stone technology to hunt and gather food. The discovery of these tools also raised important questions about the cognitive abilities and cultural practices of the Sulawesi people, and how they compared to other ancient human populations. As the research on the Sulawesi fossils and archaeology continued to unfold, it became clear that the island was a critical location for understanding the evolution and dispersal of early humans in Asia. The unique blend of modern and archaic traits in the Sulawesi fossils, combined with the rich archaeological record, made the island an important focal point for studies on human origins and migration. Dr. Chiokan's team, along with other researchers, began to explore the possibility that Sulawesi may have been a key stepping stone for early humans migrating out of Africa and into Asia, potentially interacting with other archaic groups like the Denisovans and Neanderthals. As Dr. Chiokan's team continued to study the Sulawesi fossils, they began to explore the possibility that the island may have been a key location for the interaction between early humans and other archaic groups, such as the Denisovans and Neanderthals. To further investigate this hypothesis, they collaborated with Dr. Chris Stringer, a renowned paleoanthropologist from the Natural History Museum in London, who had extensive expertise in human evolution and migration patterns. Together, they analyzed the fossil record and compared the Sulawesi fossils to other known human populations, including the Denisovans, whose presence had been detected through genetic analysis of modern humans. The comparison revealed some intriguing similarities between the Sulawesi fossils and the Denisovan genome, which suggested that there may have been interbreeding between the two groups at some point in the past. This finding sparked a new wave of interest in the study of human evolution in Asia, with several research teams embarking on expeditions to search for more fossils and archaeological evidence. One such team, led by Dr. Kira Westaway from the University of New South Wales, focused on exploring the island of Java, which was known to contain a rich fossil record of early humans. Dr. Westaway's team conducted extensive excavations at the famous Sangiran site, where they uncovered several new fossils, including a nearly complete skull of Homo erectus, a human ancestor that lived around 1.5 million years ago. The discovery provided valuable insights into the evolution and migration patterns of early humans in Asia, and it also shed light on the potential interactions between different human populations, including the Denisovans. The study of the Denisovan genome also continued to yield new insights, with researchers such as Dr. Svante Piebo from the Max Planck Institute for Evolutionary Anthropology making significant breakthroughs in the analysis of ancient DNA. By comparing the Denisovan genome to that of modern humans, Dr. Piebo and his team were able to identify specific genetic traits that had been inherited from the Denisovans, including genes related to high-altitude adaptation and immune function. These findings suggested that the Denisovans may have played a significant role in shaping the genetic diversity of modern humans, particularly in regions such as Tibet and Papua New Guinea. As Dr. Piebo's team delved deeper into the analysis of the Denisovan genome, they began to collaborate with other researchers to explore the implications of their findings on our understanding of human evolution. One such researcher was Dr. David Reich, a geneticist from Harvard University who had been studying the genetic diversity of present-day humans in Papua New Guinea and Australia. By comparing the genetic data from these populations to the Denisovan genome, Dr. Reich and his team were able to identify specific genetic traits that had been inherited from the Denisovans, including genes related to immune function and skin pigmentation. These findings suggested that the Denisovans had interbred with both Neanderthals and modern humans, leaving behind a genetic legacy that could still be seen in present-day populations. Dr. Reich's team also discovered that the Denisovan genetic material was more closely related to the genetic material of present-day humans in Papua New Guinea than it was to the genetic material of present-day humans in Europe or Asia. This suggested that the Denisovans had interbred with the ancestors of present-day Papuan populations, and that this interbreeding had occurred relatively recently in human evolutionary history. To further explore this idea, Dr. Reich's team began to study the genetic diversity of other present-day human populations in Southeast Asia and Australia. They collected DNA samples from hundreds of individuals and used advanced computational techniques to analyze the genetic data. The results of their analysis revealed a complex pattern of genetic admixture, with different populations showing evidence of interbreeding with both Denisovans and Neanderthals. One population that showed particularly high levels of Denisovan genetic material was the indigenous people of the island of Flores in Indonesia. Dr. Reich's team collected DNA samples from several individuals on the island and found that they retained a significant amount of Denisovan genetic material, including genes related to brain development and cognitive function. This discovery sparked a new wave of interest in the study of human evolution on the island of Flores, which had already been famous for the discovery of the hobbit-like species Homo floresiensis. The discovery of Homo floresiensis on the island of Flores had been made by a team of researchers led by Dr. Peter Brown from the University of New England in Australia. The team had uncovered the remains of several individuals, including a nearly complete skeleton, which they dated to around 12,000 years ago. The skeleton was remarkable for its small size, with estimates suggesting that it stood around 3 feet 7 inches tall. The discovery of Homo floresiensis had sparked a heated debate about the origins and evolution of this enigmatic species, with some researchers arguing that it represented a previously unknown species of human that had evolved in isolation on the island. Dr. Reich's discovery of high levels of Denisovan genetic material in present-day humans on the island of Flores added a new layer of complexity to this debate. It suggested that the island may have been inhabited by multiple human species, including both Homo floresiensis and the Denisovans, and that these species may have interbred with each other. To explore this idea further, Dr. Reich's team began to collaborate with Dr. Brown and his colleagues to study the genetic relationship between Homo floresiensis and the Denisovans. The team used advanced computational techniques to analyze the morphological and genetic characteristics of Homo floresiensis, comparing them to those of the Denisovans and other human species. The results of their analysis revealed a surprising pattern of similarities and differences between Homo floresiensis and the Denisovans, suggesting that these two species may have shared a common ancestor. The discovery of this common ancestor sparked a new wave of interest in the study of human evolution on the island of Flores, with researchers from around the world converging on the island to search for more fossils and genetic evidence. As the team delved deeper into the genetic relationship between Homo floresiensis and the Denisovans, they began to collaborate with other researchers who had been studying the fossil record on the island of Flores. One such researcher was Dr. Mike Morwood, an Australian archaeologist who had been instrumental in the discovery of Homo floresiensis. Dr. Morwood's team had been excavating the Liangboa cave on Flores, where the remains of Homo floresiensis had been found, and they had uncovered a wealth of new evidence that shed light on the island's complex human history. The Liangboa cave had yielded a trove of fossils, including stone tools, animal bones, and even evidence of fire use. Dr. Morwood's team had carefully dated these finds using radiometric techniques, and their results suggested that Homo floresiensis had inhabited the island from at least 100,000 years ago until around 12,000 years ago. But what was most intriguing was the presence of stone tools that seemed to predate the arrival of Homo floresiensis on the island. These tools were similar to those found in other parts of Asia, and they hinted at the possibility of an even earlier human presence on Flores. Dr. Reich's team was eager to explore this possibility further, and they began to analyze the genetic data from present-day humans on Flores to see if they could find any evidence of an earlier human migration to the island. They collected DNA samples from several individuals who lived in the vicinity of the Liangboa cave and used advanced computational techniques to compare their genetic profiles to those of other human populations in Asia. The results were striking the team, found that the present-day humans on Flores retained a small but significant amount of genetic material from an unknown human population that was distinct from both Denisovans and Homo sapiens. This unknown population seemed to have interbred with the ancestors of present-day humans on Flores, leaving behind a genetic legacy that could still be seen today. The discovery sparked a flurry of excitement among researchers who began to speculate about the identity of this mysterious human population. Some suggested that it might be related to the recently discovered red deer cave people, a previously unknown human species found in Southeast Asia. Others proposed that it could be a remnant of an even earlier human migration out of Africa, one that had occurred before the emergence of Homo sapiens. As the debate raged on, Dr. Reich's team continued to analyze the genetic data from Flores, searching for any clues that might shed light on the island's complex human history. They collaborated with other researchers who were studying the fossil record in Asia, hoping to find evidence of this mysterious human population in the archaeological record. One such researcher was Dr. Eric Trinkhaus, an American paleoanthropologist who had been studying the fossils of early humans in Asia. Dr. Trinkhaus had discovered several fossils in China and Southeast Asia that seemed to exhibit a mix of archaic and modern traits, and he had proposed that these fossils might represent a previously unknown human species. Dr. Reich's team was eager to explore this possibility further, and they began to analyze the genetic data from present-day humans in Asia to see if they could find any evidence of interbreeding between this mysterious human population and other human species. They collected DNA samples from several individuals who lived in the regions where Dr. Trinkhaus had found his fossils and used advanced computational techniques to compare their genetic profiles to those of other human populations. The results were intriguing. The team found that present-day humans in certain parts of Asia retained a small but significant amount of genetic material from this mysterious human population, suggesting that there had indeed been interbreeding between this population and other human species. The discovery opened up new avenues of research into the complex history of human evolution in Asia, and it highlighted the importance of continued exploration and analysis of the genetic and fossil records. The discovery of this mysterious human population on Flores had added a new layer of depth to the narrative, and it had raised fundamental questions about the nature of human evolution and the history of our species. As researchers delved deeper into the mystery of the Denisovans, they began to explore the possibility that this enigmatic species may have interbred with other human populations beyond Flores. Dr. Svante Pääbo, a Swedish geneticist and director of the Max Planck Institute for Evolutionary Anthropology, led a team that analyzed DNA samples from present-day humans in Papua New Guinea and Australia. They discovered that these individuals retained a significant amount of Denisovan DNA in their genomes, suggesting that there had been interbreeding between Denisovans and the ancestors of modern humans in these regions. The findings sparked a new wave of interest in the genetic history of human populations in Oceania. Dr. Pääbo's team collaborated with researchers from the University of Oxford, led by Dr. Chris Tyler Smith, to conduct a more comprehensive analysis of the genetic data. They used advanced computational methods to reconstruct the demographic history of the region and estimate the timing and extent of Denisovan admixture. The results revealed that Denisovan DNA was present in the genomes of modern humans across a wide geographic range, from the Philippines to Australia. The team estimated that the admixture had occurred around 40,000 to 50,000 years ago, when early human migrants first arrived in the region. This suggested that the Denisovans had been an established presence in Oceania for tens of thousands of years before the arrival of modern humans. To further explore the history of Denisovan-human interactions, researchers turned to the fossil record. Dr. Rainer Grun, a German paleoanthropologist, led an excavation team at the site of Liangbua on Flores, where the remains of Homo floresiensis had been discovered in 2004. The team uncovered new fossils and artifacts that provided insights into the lives of the island's ancient inhabitants. Among the finds was a well-preserved fossilized tooth, which was later analyzed by Dr. Grun's colleague, Dr. Wolfgang Muller. Using advanced scanning techniques, they were able to reconstruct the tooth's internal structure and compare it to those of other human species. The results suggested that the tooth belonged to a previously unknown human population, one that had lived on Flores before the arrival of Homo floresiensis. The discovery sparked debate among researchers about the possibility of multiple human species coexisting on the island. Dr. Grun's team proposed that the newly discovered fossils might represent a distinct species, which they termed Homo luzonensis after the nearby island of Luzon in the Philippines. The finding highlighted the complexity and diversity of human evolution in Oceania and raised new questions about the relationships between different human populations. As the search for answers continued, researchers began to explore other regions that might hold clues to the Denisovans' history. Dr. Katerina Duka, a Greek archaeologist, led an expedition to the Altai Mountains of Siberia, where the original Denisovan fingerbone had been discovered in 2008. The team conducted a thorough survey of the region's cave systems and uncovered new evidence of ancient human habitation. Among the finds was a fragment of a stone tool that showed signs of having been used by early humans around 50,000 years ago. Dr. Duka's team analyzed the tool using advanced dating techniques and confirmed its age. The discovery provided further evidence of the Denisovans' presence in the region and hinted at a more extensive human occupation of the Altai Mountains during the late Pleistocene era. As Dr. Duka's team continued to excavate the Altai Mountains, they stumbled upon a hidden cave tucked away in a remote valley. The entrance was narrow, but the team managed to squeeze through and found themselves in a vast chamber filled with ancient artifacts. Among the treasures were stone tools, fossilized bones, and mysterious symbols etched into the walls. Dr. Duka recognized some of the symbols as similar to those found in other Denisovan sites, which sparked her interest in further investigation. The team began to carefully collect and document the findings, taking care not to disturb the delicate environment. They discovered a series of hearths, indicating that early humans had used the cave for shelter and cooking. Dr. Duka's colleague, Dr. Tom Hyam, a renowned expert in radiocarbon dating, was called in to analyze the organic remains found near the hearths. His team collected samples of charcoal and bone fragments, which were then sent to the Oxford Radiocarbon Accelerator Unit for dating. The results revealed that the cave had been occupied by humans around 45,000 to 50,000 years ago during a period of significant climate change in the region. This finding was crucial, as it suggested that the Denisovans may have adapted to the changing environment in ways that allowed them to thrive in the harsh Siberian landscape. Dr. Duka's team also discovered evidence of a sophisticated tool-making industry, with intricate stone flakes and blades that showed a high degree of craftsmanship. As news of the discovery spread, other researchers began to take notice. Dr. Jean-Jacques Hublin, a French paleoanthropologist, was particularly interested in the symbolic expressions found in the cave. He saw parallels between the Altai symbols and those found in other ancient human sites, such as Blampos Cave in South Africa. Dr. Hublin proposed that these early humans may have shared a common cognitive and cultural framework, which allowed them to communicate and cooperate across vast distances. The discovery of the hidden cave in the Altai Mountains sparked a new wave of research into the lives and habits of the Denisovans. Scientists from various disciplines began to converge on the site, eager to contribute their expertise to the investigation. Dr. Svante Piebo, who had previously led the team that analyzed the Denisovan genome, became interested in the potential for ancient DNA preservation in the cave. He sent a team to collect samples of sediment and fossilized remains, which were then transported to his laboratory in Leipzig for analysis. Dr. Piebo's team began to analyze the sediment and fossilized remains collected from the Altai Mountains cave, using a combination of genetic and paleontological techniques to search for ancient DNA. They focused on the layers of sediment that corresponded to the period of Denisovan occupation, around 45,000 to 50,000 years ago. The team employed a rigorous protocol to avoid contamination, including wearing protective suits and masks and using sterile equipment to handle the samples. Meanwhile, Dr. Duka's team continued to excavate the cave, uncovering more artifacts and fossilized remains that provided valuable insights into the lives of the Denisovans. They discovered a series of personal ornaments, including pierced teeth and stone beads, which suggested a level of cognitive and cultural sophistication. The team also found evidence of a complex tool-making industry with specialized tools for tasks such as hunting and processing plant foods. As the analysis of the sediment and fossilized remains progressed, Dr. Peebo's team made a significant discovery. They identified a fragment of ancient DNA that belonged to a Denisovan individual, which they were able to reconstruct using advanced genetic techniques. The DNA fragment was remarkably well preserved, considering its age, and provided a unique opportunity to study the genetics of the Denisovans in unprecedented detail. The reconstructed DNA fragment was then compared to the previously published Denisovan genome, which had been obtained from a finger bone found in the same region. The comparison revealed a high degree of genetic similarity between the two individuals, suggesting that they belonged to the same population. However, the new DNA fragment also showed some intriguing differences, which hinted at a more complex population history than previously thought. Dr. Peebo's team decided to investigate further, using a combination of genetic and computational techniques to analyze the newly obtained DNA fragment. They collaborated with other researchers, including Dr. David Reich, a geneticist from Harvard University, who had expertise in analyzing ancient DNA. Together, they employed advanced statistical methods to reconstruct the demographic history of the Denisovans, including their population size, migration patterns, and interactions with other human groups. The analysis revealed a complex pattern of population dynamics, with evidence of gene flow between the Denisovans and both Neanderthals and modern humans. The results suggested that the Denisovans had interbred with Neanderthals in the Altai region, resulting in a significant amount of Neanderthal DNA being introduced into the Denisovan genome. Additionally, the analysis showed that the Denisovans had also interbred with modern humans, although to a lesser extent, leaving behind a genetic legacy that could still be detected in present-day human populations. The discovery of this complex population history sparked a new wave of research into the evolution and dispersal of human groups in Asia. Scientists began to re-examine the fossil record, looking for evidence of interactions between different human species. Dr. Eric Trinkhaus, a paleoanthropologist from Washington University, analyzed the fossil remains of early modern humans found in Asia, searching for signs of admixture with other human groups. Dr. Eric Trinkhaus's reanalysis of the fossil remains of early modern humans found in Asia revealed intriguing evidence of admixture with other human groups. He focused on the fossils from the Denisova cave, as well as those from other sites in China and Southeast Asia, searching for signs of interbreeding between different human species. His research suggested that early modern humans had encountered and interacted with both Neanderthals and Denisovans, leaving behind a genetic legacy that could still be detected in present-day human populations. One of the key fossils Trinkhaus examined was the 40,000-year-old remains of an early modern human found in the Tianyuan cave in China. The fossil, known as Tianyuan 1, exhibited a mix of modern and archaic traits, suggesting that it may have been the result of interbreeding between early modern humans and another human species. Trinkhaus's analysis revealed that Tianyuan 1 had a similar skull shape to modern humans, but its teeth and jaw were more robust, resembling those of Neanderthals or Denisovans. Meanwhile, Dr. David Reich and his team at Harvard University were working on a comprehensive study of ancient DNA from human fossils found in Asia. They had developed a new method for extracting DNA from fossilized remains, which allowed them to analyze the genetic material of ancient humans with unprecedented precision. The team focused on the DNA of early modern humans, Neanderthals, and Denisovans, searching for signs of gene flow and admixture between these different human groups. Reich's team made a significant breakthrough when they analyzed the DNA of a 40,000-year-old fossil found in the Baishia Karst cave in Tibet. The fossil, known as the Baishia mandible, was a jawbone that had been discovered in 1980 but had never been fully studied. Reich's team extracted DNA from the fossil and compared it to the DNA of other human fossils found in Asia. They were surprised to find that the Baishia mandible had a significant amount of Denisovan DNA, suggesting that it may have been the result of interbreeding between early modern humans and Denisovans. The discovery of Denisovan DNA in the Baishia mandible sparked a new wave of research into the history of human migration and interaction in Asia. scientists began to re-examine the fossil record, searching for evidence of Denisovan presence in other parts of the continent. Dr. Jean-Jacques Hublin, a paleoanthropologist from the Max Planck Institute, led an expedition to the Indonesian island of Sulawesi, where he had heard rumors of a mysterious human fossil that had been discovered by local villagers. Hublin's team arrived on Sulawesi and began searching for the fossil, which was said to be hidden in a remote cave deep in the jungle. After weeks of searching, they finally found the cave and recovered the fossil, which turned out to be a fragment of a human jawbone. The fossil, known as the Liang Tadongje mandible, was remarkably well preserved, with intact teeth and a clear shape that suggested it may have been from a Denisovan or a hybrid individual. Dr. Hublin's team carefully extracted the Liang Tadongje mandible from the cave and transported it to their laboratory for further analysis. The fossil was then subjected to a series of tests, including radiocarbon dating stable isotope analysis and ancient DNA extraction. The results of these tests were eagerly anticipated by the scientific community, as they had the potential to shed new light on the presence of Denisovans in Southeast Asia. Meanwhile, Dr. Reich's team at Harvard University was making progress in their analysis of the Baishia mandible. They had successfully extracted ancient DNA from the fossil and were comparing it to the DNA of other human fossils found in Asia. The preliminary results suggested that the Baishia mandible belonged to an individual who had a significant amount of Denisovan ancestry, likely as a result of interbreeding between early modern humans and Denisovans. As the news of the Liang Tadongje mandible and the Baishia mandible spread, scientists from around the world began to take notice. Dr. Chris Stringer, a paleoanthropologist from the Natural History Museum in London, was particularly interested in the findings. He had spent years studying the fossil record of human evolution in Asia and was convinced that the Denisovans played a crucial role in shaping the genetic diversity of present-day humans. Stringer decided to collaborate with Dr. Hublin and Dr. Reich to conduct a comprehensive analysis of the Liang Tadongje mandible and the Baishia mandible. Together, they assembled a team of experts from various fields, including paleoanthropology, genetics, and archaeology. The team set out to investigate the fossil record of human evolution in Asia, with a focus on the presence of Denisovans and their interactions with other human species. The team's research took them to various parts of Asia, including China, Indonesia, and the Philippines. They visited fossil sites, museums, and laboratories, collecting data and samples that would help them reconstruct the history of human migration and interaction in the region. One of the key sites they visited was the cave system in Laos, where the fossil remains of early modern humans had been found in association with stone tools and other archaeological artifacts. The Laotian cave system, known as the Tamapaling Cave, had been excavated by a team of archaeologists from the University of Illinois. The excavation had uncovered a wealth of fossils and artifacts, including stone tools, bone fragments, and ancient hearths. Dr. Stringer's team was particularly interested in the fossil remains found at the site, which included several teeth and jawbones that appeared to be from early modern humans. The team spent several weeks analyzing the fossils and artifacts from the Tamapaling Cave, using a range of techniques including radiocarbon dating, stable isotope analysis, and ancient DNA extraction. The results of their analysis suggested that the early modern humans who lived in the region had interacted with other human species, including the Denisovans and possibly even the Neanderthals. As the team delved deeper into their research, they began to uncover a complex picture of human evolution and migration in Asia. It became clear that the region had been inhabited by a diverse range of human species, each with their own unique characteristics and adaptations. The team's findings challenged the traditional view of human evolution, which had long been dominated by the out-of-Africa hypothesis. The out-of-Africa hypothesis proposed that early modern humans had originated in Africa and then migrated to other parts of the world, replacing other human species such as the Neanderthals and Denisovans. However, Dr. Stringer's team found evidence that suggested a more complex scenario with multiple waves of migration and interaction between different human species. As Dr. Stringer's team continued to analyze the fossils and artifacts from the Tamapaling Cave, they began to notice a peculiar pattern of genetic exchange between early modern humans and Denisovans. The ancient DNA extracted from the fossils revealed a significant amount of Denisovan ancestry in the early modern human population, suggesting that there had been extensive interbreeding between the two species. This finding was further supported by the discovery of a similar genetic pattern in present-day populations of Southeast Asia, particularly in Papua New Guinea and Australia. The team's research also shed light on the migratory patterns of early modern humans and Denisovans. It appeared that early modern humans had migrated to Asia through multiple routes, including the southern coastal route and the northern inland route. The southern coastal route would have taken them along the coast of India and Southeast Asia, while the northern inland route would have taken them through the Middle East and Central Asia. The Denisovans, on the other hand, seem to have been limited to a more restricted geographic range, with their presence mostly confined to Southeast Asia and Australia. One of the key researchers who contributed to this understanding was Dr. Svante Pääbo, a Swedish geneticist who had pioneered the field of ancient DNA analysis. Pääbo's team at the Max Planck Institute for Evolutionary Anthropology in Leipzig, Germany, had been instrumental in extracting and analyzing ancient DNA from fossil remains, including those of the Denisovans. Their research had provided crucial insights into the genetic relationships between different human species and had helped to reconstruct the history of human migration and interaction. As the team delved deeper into their research, they began to explore the archaeological record of Southeast Asia in greater detail. They visited sites such as the Naya Cave in Malaysia and the Tabon Cave in the Philippines, where fossil remains of early modern humans and other human species had been found. The team's analysis of the archaeological evidence revealed a complex picture of human occupation and migration in the region, with multiple waves of migration and interaction between different human species. The discovery of stone tools and other artifacts at these sites also provided valuable insights into the technological and cultural capabilities of early modern humans and Denisovans. For example, the presence of sophisticated stone tools at the Naya Cave suggested that early modern humans had developed advanced technological capabilities, while the presence of more primitive tools at the Tabon Cave suggested that the Denisovans may have had more limited technological abilities. The team's research also highlighted the importance of climate change in shaping human migration and interaction patterns. The region's climate had undergone significant changes over the past 100,000 years, with periods of glaciation and warming affecting the availability of resources and habitats for different human species. For example, during the last ice age, much of Southeast Asia would have been covered in dense rainforest, making it difficult for humans to migrate through the region. As the team delved deeper into the impact of climate change on human migration patterns, they turned their attention to the work of Dr. Christopher Turney, a British climatologist who had spent years studying the paleoclimate of Southeast Asia. Turney's research had shown that the region's climate had undergone significant fluctuations over the past 100,000 years, with periods of severe drought and flooding affecting the availability of resources and habitats for different human species. The team was particularly interested in Turney's findings on the role of the Indonesian throughflow, a warm ocean current that flows through the Indonesian archipelago and plays a crucial role in shaping the region's climate. The Indonesian throughflow, Turney had discovered, had been significantly weaker during the last ice age, resulting in a drier and cooler climate in Southeast Asia. This, in turn, would have made it more difficult for humans to migrate through the region and may have even led to the isolation of certain populations, such as the Denisovans. The team realized that this could have significant implications for their understanding of human evolution and migration patterns, and they began to explore the possibility that the Indonesian throughflow had played a key role in shaping the genetic diversity of present-day humans. To further investigate this hypothesis, the team turned to the work of Dr. Peter Bellwood, a British archaeologist who had spent years studying the prehistory of Southeast Asia. Bellwood's research had shown that the region had been inhabited by a diverse range of human populations, each with their own unique cultural and technological traditions. The team was particularly interested in Bellwood's findings on the spread of Austronesian languages, which are spoken by many present-day populations in Southeast Asia and the Pacific. Bellwood's research had shown that the Austronesian language family had originated in Taiwan around 6,000 years ago and had then spread rapidly throughout Southeast Asia and the Pacific. The team realized that this could have significant implications for their understanding of human migration patterns, and they began to explore the possibility that the spread of Austronesian languages had been driven by the migration of farmers and sailors who had brought new technologies and cultural practices to the region. As they delved deeper into the history of Southeast Asia, the team encountered the work of Dr. Philip Piper, a British archaeologist who had spent years excavating sites in the Philippines and Malaysia. Piper's research had shown that the region had been inhabited by sophisticated human populations with complex social hierarchies and technological traditions. The team was particularly interested in Piper's findings on the presence of early pottery and metalworking technologies in the region, which suggested that human populations in Southeast Asia had been highly adaptable and innovative. The discovery of these technologies also raised important questions about the relationship between human migration patterns and the spread of cultural and technological innovations. Had the Denisovans, for example, played a role in the development of these technologies, or had they been isolated from other human populations? The team realized that answering these questions would require a more detailed analysis of the archaeological record, and they began to plan a new series of excavations and field surveys in Southeast Asia. The team's plans for a new series of excavations and field surveys in Southeast Asia led them to the island of Luzon in the Philippines, where Dr. Philip Piper had been conducting research on the archaeology of the region. They joined forces with Piper and his team, who were investigating the Eel Cave site, a limestone cave that had yielded a wealth of archaeological evidence, including stone tools, pottery, and human remains. The team was particularly interested in the possibility that the Eel Cave site might contain evidence of Denisovan presence given its location in the northern part of Luzon, which was thought to have been a potential migration route for early humans. As they began their excavation, the team encountered a complex stratigraphy with multiple layers of sediment and archaeological deposits. They carefully excavated each layer, documenting every find and collecting samples for further analysis. The site yielded a range of artifacts, including stone tools such as flake scrapers and hammerstones, as well as pottery shards and other ceramic remains. The team also found evidence of human activity, including hearths and cooking pits, which suggested that the cave had been occupied by humans for extended periods. One of the most significant discoveries at the Eel Cave site was the presence of a previously unknown type of stone tool, which the team dubbed the Luzon flake. This tool was characterized by its distinctive shape and edge morphology, which was unlike anything found at other sites in Southeast Asia. The team suspected that the Luzon flake might be associated with the Denisovans, given its unique characteristics and the fact that it was found in a context that suggested a relatively old age. To further investigate the origins of the Luzon flake, the team turned to the expertise of Dr. Huey Tieng Wah, a Malaysian archaeologist who had spent years studying the stone tool traditions of Southeast Asia. Wah's research had shown that the region's stone tools were highly diverse, with different types and styles associated with different human populations. The team was interested in learning more about the technological and cultural context in which the Luzon flake had been produced, and Wah's expertise proved invaluable in helping them to understand the significance of this discovery. As they delved deeper into the analysis of the Eel Cave site, the team also encountered the work of Dr. Elizabeth Matasu-Smith, a New Zealand geneticist who had been studying the genetics of human populations in Southeast Asia. Matasu-Smith's research had shown that the region's humans were highly genetically diverse, with different populations showing distinct genetic signatures. The team was interested in learning more about the genetic relationships between the humans who had occupied the Eel Cave site and other populations in Southeast Asia, and Matasu-Smith's expertise proved crucial in helping them to understand the complex genetic landscape of the region. As the team delved deeper into the analysis of the Eel Cave site, they began to explore the genetic relationships between the humans who had occupied the site and other populations in Southeast Asia. Dr. Elizabeth Matasu-Smith's expertise proved invaluable in helping them understand the complex genetic landscape of the region. She explained that the genetics of human populations in Southeast Asia were shaped by a combination of factors, including migration, admixture, and isolation. The team was particularly interested in learning more about the genetic legacy of the Denisovans, and how it had contributed to the genetic diversity of present-day humans in the region. To shed more light on this topic, the team turned to the study of ancient DNA. They worked with Dr. Svante Pääbo, a Swedish geneticist who had pioneered the field of ancient DNA research. Pääbo's team had developed sophisticated techniques for extracting and analyzing DNA from ancient human remains, including those found in Southeast Asia. The team collaborated with Pääbo to analyze the DNA of several ancient human individuals found in the region, including those from the Ile cave site. The results of the analysis were striking. The team found that many present-day humans in Southeast Asia retained a small amount of Denisovan DNA in their genomes, suggesting that there had been interbreeding between Denisovans and modern humans in the past. This was consistent with previous studies that had found similar patterns of admixture in other parts of the world, such as Papua New Guinea and Australia. The team also found that the genetic legacy of the Denisovans was more pronounced in certain populations, such as the indigenous groups of the Philippines and Malaysia. As the team continued to explore the genetics of human populations in Southeast Asia, they began to uncover a complex pattern of migration and admixture that had shaped the region's history. They worked with Dr. Mark Stoneking, an American geneticist who had studied the genetics of human migration in the region. Stoneking's research had shown that the initial peopling of Southeast Asia had occurred around 50,000 to 60,000 years ago, when modern humans first migrated out of Africa and into the region. The team found that this initial migration had been followed by subsequent waves of migration, including the arrival of Austronesian-speaking peoples from Taiwan around 4,000 to 5,000 years ago. The team's research also highlighted the importance of the Sunda Shelf, a region of shallow water and coastal plains that had connected the islands of Southeast Asia during periods of low sea level. The Sunda Shelf had played a crucial role in facilitating human migration and exchange between different parts of the region, and it helped to shape the genetic diversity of present-day humans in Southeast Asia. As the team continued to explore the history of human migration and admixture in the region, they began to uncover a rich and complex narrative that highlighted the dynamic and interconnected nature of human populations in Southeast Asia. The study of ancient DNA and genetics was complemented by archaeological research, which provided valuable insights into the cultural and technological practices of ancient humans in the region. The team worked with Dr. Peter Bellwood, a British archaeologist who had studied the prehistory of Southeast Asia. Bellwood's research had shown that the region had been characterized by a series of complex societies, including the Neolithic cultures of the Philippines and Malaysia, which had developed sophisticated technologies and social hierarchies. As the team delved deeper into the archaeological record, they began to uncover evidence of cultural exchange and diffusion between different parts of Southeast Asia. They found that certain artifacts, such as pottery and metal tools, had been traded and exchanged across the region, reflecting a high degree of connectivity and interaction between ancient human populations. The team also discovered that certain cultural practices, such as rice agriculture and animal husbandry, had been introduced to the region from outside, highlighting the dynamic and adaptive nature of human societies in Southeast Asia. As the team continued to unravel the complexities of human migration and cultural exchange in Southeast Asia, they turned their attention to the Neolithic period, which saw the introduction of new technologies and practices that would have a profound impact on the region's population. One of the key figures in this area of research was Dr. Charles Hyam, a British archaeologist who had spent decades studying the prehistory of Southeast Asia. Hyam's work had focused on the Ban Non Wat site in Thailand, which had yielded a wealth of information about the lives of Neolithic farmers in the region. The team worked with Hyam to analyze the archaeological evidence from Ban Non Wat, which included elaborate pottery, bronze tools, and evidence of rice agriculture. They found that the Neolithic period in Southeast Asia was characterized by a high degree of cultural complexity, with societies organized into hierarchical chiefdoms and engaging in extensive trade networks. The team also discovered that the Neolithic farmers of Ban Non Wat had a sophisticated understanding of astronomy, as evidenced by the alignment of their burial mounds with celestial bodies. As they delved deeper into the archaeology of the region, the team encountered the work of Dr. Wilhelm Solheim, an American anthropologist who had spent years studying the prehistory of Southeast Asia. Solheim's research had focused on the Nusantau maritime trading network, which had connected the islands of Southeast Asia and beyond from around 5000 to 1000 BCE. The team found that this network had played a crucial role in the dissemination of cultural practices, technologies, and ideas across the region, and had helped to shape the genetic diversity of present-day humans in Southeast Asia. The team's research also took them to the island of Luzon in the Philippines, where they worked with Dr. Alfred Paulich, a German archaeologist who had discovered evidence of a previously unknown Neolithic culture. The site, known as Ifugao, had yielded a range of artifacts, including pottery, stone tools, and evidence of rice agriculture. The team found that the Ifugao culture was characterized by a unique blend of Austronesian and indigenous influences, reflecting the complex history of human migration and cultural exchange in the region. As they continued to explore the archaeology of Southeast Asia, the team encountered the work of Dr. Peter Lape, an American archaeologist who had studied the prehistory of the Indonesian archipelago. Lape's research had focused on the island of Sulawesi, where he had discovered evidence of a previously unknown Neolithic culture. The team found that this culture was characterized by a sophisticated understanding of metallurgy, as evidenced by the presence of bronze and iron tools at archaeological sites across the island. The team's research in Sulawesi also took them to the site of Lake Matano, where they worked with Dr. Rainer Grun, an Australian geologist who had studied the region's geological history. Grun's research had shown that the lake had been formed as a result of tectonic activity around 2 million years ago and had since become a focal point for human habitation in the region. The team found that the lake's shores were dotted with archaeological sites, including evidence of Neolithic farming and metalworking. As the team delved deeper into the archaeology of Sulawesi, they became increasingly fascinated by the island's unique cultural and geological history. Dr. Grun's research on Lake Matano had provided valuable insights into the region's tectonic activity, and the team was eager to explore the lake's shores further. They spent several weeks conducting excavations at the lake's edge, uncovering evidence of Neolithic farming and metalworking that dated back thousands of years. One of the most significant discoveries made during this period was the finding of an ancient bronze axe head, which was later dated to around 2000 BCE. The axe head was remarkably well preserved, with intricate designs and patterns etched into its surface. The team was amazed by the level of craftsmanship and sophistication that had gone into creating such an object, and they realized that the Neolithic culture of Sulawesi was far more advanced than they had initially thought. As they continued to explore the lake shores, the team encountered the work of Dr. David Balbeck, an Australian archaeologist who had spent years studying the prehistory of Southeast Asia. Balbeck's research had focused on the spread of Austronesian languages and cultures across the region, and he had developed a number of theories about the migration patterns of ancient populations. The team was particularly interested in Balbeck's work on the genetic origins of the Sulawesi population, which suggested that the island had been inhabited by a complex mixture of indigenous and migrant groups. They spent several weeks discussing and debating Balbeck's theories, and they began to develop their own ideas about the cultural and genetic history of the region. One of the key questions that the team was trying to answer was how the Denisovan DNA had ended up in the modern human population of Southeast Asia. They knew that the Denisovans had interbred with both Neanderthals and modern humans, but they were not sure when or where this interbreeding had occurred. They spent many hours poring over genetic data, looking for clues about the timing and location of these ancient encounters. As they delved deeper into the genetic data, the team began to notice a number of interesting patterns and anomalies. They found that the Denisovan DNA was not evenly distributed across the modern human population of Southeast Asia, but was instead concentrated in certain regions and communities. They also discovered that the Denisovan DNA was often associated with specific genetic traits and characteristics, such as dark skin and curly hair. The team realized that these findings had significant implications for their understanding of human evolution and migration patterns. They began to develop a new theory about the origins of the Denisovan DNA in Southeast Asia, one that suggested that the Denisovans had played a far more significant role in shaping the genetic landscape of the region than they had initially thought. As they continued to refine and develop their theory, the team encountered the work of Dr. Eske Willerslev, a Danish geneticist who had spent years studying the ancient DNA of human populations. Willerslev's research had focused on the genetics of ancient Siberian populations, and he had made a number of groundbreaking discoveries about the origins of modern humans in Asia. The team was particularly interested in Willerslev's work on the ancient DNA of the Altai Mountains, which had revealed a complex pattern of genetic exchange and interbreeding between different human populations. They spent several weeks discussing and debating Willerslev's findings, and they began to see parallels between the genetic history of Siberia and the genetic history of Southeast Asia. As they explored these parallels further, the team began to develop a new understanding of the ancient migration patterns that had shaped the genetic landscape of Asia. They realized that the Denisovans had been just one part of a complex and dynamic system of human populations, one that had stretched from Siberia to Southeast Asia and had involved numerous episodes of interbreeding and genetic exchange. As the team delved deeper into the genetic history of Asia, they became increasingly fascinated by the work of Dr. Svante Pääbo, a Swedish geneticist who had made groundbreaking discoveries about the Denisovans and their interactions with modern humans. Pääbo's research had focused on the analysis of ancient DNA from fossils found in the Denisova cave in Siberia, which had yielded a wealth of information about the genetics and evolution of the Denisovans. One of the key findings to emerge from Pääbo's research was the discovery that the Denisovans had interbred with both Neanderthals and modern humans, leaving behind a genetic legacy that could still be seen in the DNA of present-day populations. The team was particularly interested in the fact that the Denisovan DNA was found to be more closely related to the DNA of modern humans from Papua New Guinea than to that of modern humans from other parts of the world. This finding suggested that there had been a previously unknown migration of modern humans out of Africa, one that had taken them through Asia and into the Pacific, where they had encountered and interbred with the Denisovans. The team spent many hours discussing the implications of this discovery, and they began to develop new theories about the migration patterns and genetic history of early human populations. As they explored these theories further, the team turned to the work of Dr. Johannes Krauss, a German geneticist who had made significant contributions to the field of ancient DNA analysis. Krauss's research had focused on the development of new methods for extracting and analyzing ancient DNA from fossil remains, and he had played a key role in the discovery of the Denisovan genome. The team was particularly interested in Krauss's work on the genetic history of the Aboriginal Australian population, which had revealed a complex pattern of genetic exchange and interbreeding between different human populations. They spent several weeks discussing Krauss's findings, and they began to see parallels between the genetic history of Australia and the genetic history of Papua New Guinea. As they explored these parallels further, the team became increasingly fascinated by the work of Dr. Peter Bellwood, a British archaeologist who had made significant contributions to our understanding of human migration and cultural exchange in the Pacific. Bellwood's research had focused on the analysis of archaeological and linguistic data from the region, and he had developed a number of theories about the origins and spread of Austronesian languages and cultures. The team was particularly interested in Bellwood's work on the Lapita culture, which had emerged in the Pacific around 3,500 years ago and had played a key role in the spread of Austronesian languages and cultures throughout the region. They spent many hours discussing Bellwood's findings, and they began to see connections between the genetic history of the Pacific and the cultural and linguistic history of the region. As the team delved deeper into Bellwood's work on the Lapita culture, they became fascinated by the similarities between the archaeological findings in the Pacific and the genetic data from the region. The Lapita people, known for their distinctive pottery and seafaring skills, had spread Austronesian languages and cultures throughout the Pacific, leaving behind a trail of genetic and cultural markers that could still be seen today. One of the key sites that provided insight into the Lapita culture was the Talasu site on the island of Iefit in Vanuatu, where archaeologists had uncovered evidence of early Lapita settlement dating back to around 3,000 years ago. The team spent several weeks studying the archaeological findings from Talasiu, including the discovery of pottery shards and stone tools that were characteristic of the Lapita culture. They also analyzed the genetic data from modern-day populations in Vanuatu, which showed a clear connection to the Austronesian-speaking peoples of Taiwan and the Philippines. This led them to investigate the work of Dr. Marie Lin, a Taiwanese geneticist who had made significant contributions to our understanding of the genetic history of Austronesian-speaking peoples. Dr. Lin's research had focused on the analysis of mitochondrial DNA from modern-day populations in Taiwan and the Philippines, which had revealed a complex pattern of genetic exchange and migration between different groups. The team was particularly interested in her work on the Amis people of Taiwan, who had been found to have a high frequency of a specific mitochondrial DNA haplogroup that was also common among the Lapita people. This suggested that there may have been a previously unknown migration of people from Taiwan to the Pacific, which could have played a key role in the spread of Austronesian languages and cultures. As the team explored this idea further, they turned to the work of Dr. Catherine Wellington, a New Zealand-based archaeologist who had made significant contributions to our understanding of the settlement of the Pacific. Dr. Wellington's research had focused on the analysis of archaeological data from sites in New Zealand and the surrounding islands, which had revealed a complex pattern of migration and cultural exchange between different groups. The team was particularly interested in her work on the Moriori people, who had lived on the Chatham Islands off the coast of New Zealand and had been found to have a unique genetic profile that was distinct from other Pacific populations. The discovery of the Moriori people's unique genetic profile had sparked a lively debate among researchers about the origins and history of the Pacific. Some had suggested that the Moriori may have been a remnant population of an earlier wave of migration to the Pacific, while others had argued that they may have been the result of a later migration from New Zealand. The team spent several weeks discussing these different theories and analyzing the available data, which led them to investigate the work of Dr. Lisa Matisu Smith, a Canadian geneticist who had made significant contributions to our understanding of the genetic history of Pacific populations. Dr. Lisa Matisu Smith's research on the genetic history of Pacific populations had focused on the analysis of ancient DNA from archaeological sites throughout the region. Her work had revealed a complex pattern of migration and admixture between different groups, which had shed new light on the settlement of the Pacific. The team was particularly interested in her study on the ancient DNA of the Rapa Nui people, who had lived on Easter Island and had been found to have a unique genetic profile that was distinct from other Pacific populations. Dr. Matisu Smith's research had provided new insights into this debate, and the team spent several weeks analyzing her data and discussing the implications. One of the key findings of Dr. Matisu Smith's research was the presence of a specific genetic marker that was common among the Rapa Nui people, but rare in other Pacific populations. This marker had been found to be associated with the indigenous peoples of South America, and its presence on Easter Island suggested that there may have been contact between the Rapa Nui and South American populations at some point in the past. The team was fascinated by this discovery and decided to investigate further. They turned to the work of Dr. Erik Thorsby, a Norwegian geneticist who had made significant contributions to our understanding of the genetics of indigenous peoples in South America. Dr. Thorsby's research had focused on the analysis of genetic data from modern-day populations in Chile and Argentina, which had revealed a complex pattern of migration and admixture between different groups. The team was particularly interested in his work on the Mapuche people, who had been found to have a unique genetic profile that was distinct from other indigenous populations in South America. The Mapuche people had lived in the region of southern Chile and Argentina for thousands of years, and their genetic profile reflected a complex history of migration and admixture. Dr. Thorsby's research had revealed that the Mapuche people had a high frequency of a specific genetic marker that was also common among the Rapa Nui people. This suggested that there may have been contact between the Mapuche and Rapa Nui populations at some point in the past, and the team decided to investigate further. They spent several weeks analyzing Dr. Thorsby's data and discussing the implications of his research. The discovery of a genetic link between the Mapuche and Rapa Nui people had significant implications for our understanding of the settlement of the Pacific and the history of contact between different populations. As they delved deeper into the research, they came across the work of Dr. David Reich, a Harvard geneticist who had made significant contributions to our understanding of the genetics of ancient human populations. Dr. Reich's research had focused on the analysis of ancient DNA from archaeological sites throughout the world, including the Denisova cave in Siberia, where the fingerbone of the Denisovan girl had been discovered. The team was particularly interested in his work on the genetic legacy of the Denisovans and their contribution to the modern human gene pool. Dr. Reich's research had revealed that the Denisovans had interbred with both Neanderthals and modern humans, leaving behind a genetic legacy that could still be seen today. The team's investigation led them to the island of New Guinea, where they found evidence of a complex pattern of migration and admixture between different groups. The indigenous peoples of New Guinea had been found to have a high frequency of Denisovan DNA, which suggested that there may have been contact between the Denisovans and modern human populations in the region at some point in the past. The team spent several weeks analyzing the genetic data from New Guinea and discussing the implications of their research. Dr. Stringer's research had focused on the analysis of fossil evidence from archaeological sites throughout the world, including the discovery of Homo floresiensis in Indonesia. The team was particularly interested in his work on the origins of modern humans and the role of the Denisovans in human history. Dr. Stringer's work on Homo floresiensis had shed light on the complex history of human evolution in Asia, and the team was eager to learn more about the relationship between this enigmatic species and the Denisovans. They delved into the research of Dr. Peter Brown, an Australian anthropologist who had led the team that discovered Homo floresiensis on the island of Flores in 2004. The discovery of this diminutive human species, which lived as recently as 12,000 years ago, had raised questions about the origins of the Denisovans and their potential interactions with other human species. As they explored the fossil record, the team came across the work of Dr. Katerina Doka, a Greek archaeologist who had worked on the excavation of the Denisova cave in Siberia. Dr. Doka's research had focused on the archaeological context of the cave, including the discovery of stone tools and other artifacts that suggested a complex history of human occupation. The team was fascinated by the possibility that the Denisovans may have coexisted with other human species, such as Neanderthals and modern humans, and they spent several weeks analyzing the archaeological evidence from the cave. The team's investigation led them to the work of Dr. Svante Pääbo, a Swedish geneticist who had made significant contributions to our understanding of ancient DNA. Dr. Pääbo's research had focused on the analysis of DNA from fossil remains, including the fingerbone of the Denisovan girl found in the Denisova cave. The team was amazed by the breakthroughs that had been achieved in the field of ancient DNA analysis and the insights it had provided into human evolution and migration. Dr. Krause's research had focused on the genetic legacy of the Denisovans and their contribution to the modern human gene pool. The team spent several weeks analyzing the genetic data and discussing the implications of Dr. Krause's research, including the possibility that many present-day humans retain a small amount of Denisovan DNA in their genomes. Dr. Willerslev's research had focused on the analysis of DNA from fossil remains found in Asia, including the discovery of a 40,000-year-old human jawbone in Laos that contained a mix of modern human and Denisovan DNA. The team was fascinated by the implications of this discovery and spent several weeks discussing the potential interactions between different human species in the region. The team's investigation led them to the work of Dr. Robin Donnell, a British archaeologist who had worked on the excavation of ancient human sites in Asia. The team spent several weeks analyzing the archaeological evidence and discussing the implications of Dr. Donnell's research, including the possibility that the Denisovans may have played a significant role in shaping the course of human evolution in Asia. Dr. Petralia's work focused on the discovery of stone tools and other artifacts in the Arabian Peninsula, which suggested that early humans, including possibly the Denisovans, had migrated out of Africa and into Asia via the southern route. The team was fascinated by the implications of this research, as it provided evidence of a complex network of human migration and interaction that stretched across the continent. Dr. Petraglia's research also highlighted the importance of the Arabian Peninsula as a crossroads for early human migration. The team learned about the discovery of ancient stone tools in the region, which dated back to around 125,000 years ago, and the presence of fossil remains of early humans, such as Homo erectus. The team spent several weeks analyzing the archaeological evidence from the region, including the excavation of sites such as the Jebel Faya rock shelter in the United Arab Emirates. The team's investigation also led them to the work of Dr. Hans-Peter Erpmann, a German archaeologist who had conducted research on the early human settlement of the Middle East. Dr. Erpmann's work focused on the discovery of ancient stone tools and other artifacts in the Levant region, which suggested that early humans had migrated into the region from Africa and interacted with other human species, such as the Neanderthals. They turned to the work of Dr. David Reich, an American geneticist who had conducted extensive research on the genetics of ancient humans. The team learned about the discovery of a distinctive genetic signature in present-day humans, which suggested that many people retain a small amount of Denisovan DNA in their genomes. The team spent several weeks analyzing the genetic data and discussing the implications of Dr. Reich's research. They explored the possibility that the Denisovans had interbred with other human species, such as modern humans and Neanderthals, and that this interbreeding had left a lasting legacy in the genetic makeup of present-day humans. The team's investigation also led them to the work of Dr. Pontus Skoglund, a Swedish geneticist who had conducted research on the genetics of ancient humans. As the team delved deeper into the genetic legacy of the Denisovans, they began to explore the work of Dr. Svante Pääbo, a Swedish geneticist who had conducted extensive research on the DNA of ancient humans. Dr. Pääbo's work at the Max Planck Institute for Evolutionary Anthropology had been instrumental in the discovery of the Denisovan genome, and his team had made significant contributions to the field of paleogenetics. The team learned about the challenges of extracting DNA from fossil remains, particularly those that were tens of thousands of years old, and how Dr. Pääbo's team had developed innovative methods to overcome these challenges. One of the key breakthroughs in the study of Denisovan genetics came with the discovery of a finger bone in the Denisova cave, which was found to contain exceptionally well-preserved DNA. The team analyzed the DNA from this finger bone, as well as from a tooth found in the same cave, and compared it to the DNA of modern humans and Neanderthals. The team also explored the work of Dr. Joshua Akey, an American geneticist who had conducted research on the genetics of archaic humans, including the Denisovans. Dr. Akey's work had focused on the analysis of DNA from fossil remains found in Southeast Asia, and his team had made significant contributions to our understanding of the migration patterns of early humans. The team learned about the discovery of a 40,000-year-old human skeleton in the Philippines, which was found to contain a mix of modern human and Denisovan DNA. As the team continued to explore the genetics of the Denisovans, they began to examine the implications of this research for our understanding of human evolution. They turned to the work of Dr. Chris Stringer, a British anthropologist who had conducted extensive research on the evolution of humans. Dr. Stringer's work had focused on the fossil record of human evolution, and his team had made significant contributions to our understanding of the migration patterns of early humans. The team learned about the discovery of fossils in Africa, Asia, and Europe, which provided evidence of a complex pattern of human migration and interaction that stretched across the globe. The team also explored the work of Dr. Rebecca Ackerman, a South African anthropologist who had conducted research on the evolution of human physiology. Dr. Ackerman's work had focused on the analysis of fossil remains found in Africa, and her team had made significant contributions to our understanding of the adaptations that allowed early humans to thrive in a variety of environments. The team learned about the discovery of fossils in South Africa, which provided evidence of a complex pattern of human evolution that was influenced by a range of factors, including climate, diet, and geography. As the team delved deeper into the genetics of the Denisovans, they began to collaborate with other researchers to integrate their findings with the broader context of human evolution. Dr. Stringer's work on the fossil record of human evolution was complemented by the genetic analysis of Dr. Pääbo's team, providing a more comprehensive understanding of the complex interactions between different human populations. The discovery of Denisovan DNA in modern humans in Southeast Asia and Australia sparked a new wave of research into the migration patterns and admixture events that had shaped the genetic landscape of these regions. Dr. Akey's work on the genetics of archaic humans, including the Denisovans, was instrumental in developing new methods for analyzing ancient DNA. His team's research focused on the development of more sensitive and efficient techniques for extracting and sequencing DNA from fossil remains, which enabled the analysis of smaller and more degraded samples. This breakthrough led to a significant increase in the number of ancient human genomes that could be studied, providing valuable insights into the evolutionary history of our species. The integration of genetic and fossil evidence also shed new light on the cognitive and cultural abilities of the Denisovans. Dr. Ackerman's research on human physiology and evolution highlighted the importance of considering the interplay between genetic and environmental factors in shaping human adaptations. The discovery of sophisticated stone tools and personal ornamentation at Denisovan sites, such as the cave in Siberia where the finger bone was found, suggested that these archaic humans may have possessed a level of cognitive and cultural complexity similar to that of modern humans. As the study of the Denisovans continued to advance, researchers began to explore the potential implications of their findings for our understanding of human health and disease. Dr. Skoglund's work on the genetics of ancient humans had shown that the introgression of Neanderthal and Denisovan DNA into the modern human gene pool had contributed to the evolution of certain traits, such as skin color and immune function. This raised important questions about the potential role of archaic human DNA in shaping our susceptibility to diseases and our response to environmental stressors. The collaboration between geneticists, anthropologists, and archaeologists working on the Denisovan project led to a significant expansion of our knowledge of human evolution and migration patterns. The discovery of Denisovan DNA in modern humans had sparked a new wave of research into the complex interactions between different human populations, and the integration of genetic and fossil evidence had provided a more comprehensive understanding of the evolutionary history of our species. As researchers continued to analyze the wealth of data generated by the Denisovan project, they began to uncover new insights into the lives and experiences of these enigmatic, archaic humans and their place within the broader narrative of human evolution. The study of the Denisovans also highlighted the importance of preserving and protecting archaeological sites, particularly those that contain fossil remains or other evidence of ancient human activity. The destruction or degradation of these sites could result in the loss of irreplaceable information about our evolutionary past and the denial of future generations access to this knowledge. In response to this concern, researchers and conservationists began to work together to develop new strategies for protecting and preserving archaeological sites, and to raise awareness about the importance of preserving our cultural and evolutionary heritage. As the research community continued to explore the many facets of Denisovan biology and culture, they were reminded of the profound impact that these archaic humans had on the course of human evolution. The discovery of their DNA in modern humans had opened up new avenues for understanding the complex history of our species, and the integration of genetic and fossil evidence had provided a more nuanced and detailed picture of the lives and experiences of these ancient humans. The study of the Denisovans would continue to be an active and vibrant area of research, driven by the ongoing discovery of new fossils, artifacts, and genetic data, and fueled by the enduring fascination with our evolutionary past. Their genetic legacy lives on within us, a testament to the complex and interconnected history of our species. The discovery of their existence has not only expanded our understanding of the past, but has also invited us to reconsider our place within the grand narrative of human history. Thank you for joining me throughout this historical journey. That's all for today's History with Herodotus. Remember to like, share, and subscribe. And until next time, remember that to understand the present, we must first understand the past.